Hello everyone, so I'm going to talk about the molecular characterization of canine mammary tumors that were collected and analyzed as a part of the UMass Canine Tumor Project. So we chose to study canine tumors specifically because canine cancer is actually a really good model for human breast cancer. Environment plays a really big role in the development of cancer, and both humans and dogs do share the same environment. Both canine and human mammary tumors also occur naturally or spontaneously, so there's no need to induce the tumor as you would if you used a rodent model. So tumors that do develop in the canine mammary glands have altered gene expression of many of the same genes that are also altered in human breast tumors. And treatment for cancer is also very similar for both dogs and humans. Some of the same treatments in dogs work in humans to similar degrees, and one goal is that in the future we'll be able to test additional treatments on dogs to then be used for humans. So just a couple of additional facts are that 52% of all tumors in female dogs are mammary tumors, as compared to just 13% in women. It's the most common cancer type in canines and the most common cancer type in women, second to skin cancer. And of the mammary tumors that do develop in canines, 50% of them are malignant and the other 50% are benign or non-harmful. In comparison to humans, only about 20% of the lumps found in the breast are malignant. So to start this project, we reached out to over 50 veterinarians, asking them to send us tumors that they removed as a part of their routine surgeries. And each vet was sent a kit that you can see on the far left with all the necessary instructions for them to ship the tumor back to us in a sterile container containing a saline buffer solution. And the tumors were shipped back in an overnight FedEx box with a prepaid label. And if we were not able to have access to the tumor right away, we were still able to process the tumor days later. So once we received the actual tumors, they were cut into 5 pieces to be used for nucleic acid extraction, patient-derived xenografts, tissue staining, and cell culture. The main way that we used the tumors was for tissue analysis, cell line development, and gene expression comparison among those cell lines. The tumor pieces that are going to be used for nucleic acid extraction were flash frozen, and those that are going to be used for PDX were cryopreserved. So the tumors we received had a large variation in breeds that they came from, as well as tumor type. We took the cells from each tumor to develop a cell line specific to each tumor, and these cell lines are called UMACT109, 111, and 112. Below each cell line name, you can see the breed of dog that it came from, as well as the tumor type that it was. And there are higher risks of tumor development in older dogs, as well as small breed dogs. Also, if the dog is purebred or not spayed, this further increases their chance of mammary tumor development. All of these dogs are considered to be older and smaller breed dogs, and the dogs whose tumors were used for 111 and 112 were also purebred dogs. So these are actual pictures from each of our cell lines, and in humans, it's almost always the epithelial cells of the breast that form the cancer, but in canines, the stromal cells of the supporting tissue in the mammary glands also commonly form the tumors. UMACT109 is very stromal in appearance, meaning that the cells are very long, and that's why they kind of have that stretched out look. The cells in 111 are very classically epithelial and look more square and have a cuboidal shape, and they have those very defined edges. And 112 appears to have a mix of both stromal and epithelial cells, because some cells have that longer, more stretched out appearance, and some are very cuboidal and classically epithelial looking. So hematoxylin and eosin staining is one of the first things pathologists look at when determining if a tissue is possibly cancerous. To do this, the tissue is placed in a 37% formaldehyde solution, then encased in a paraffin wax, and then thinly sliced to show just a cross-section of the tumor, and is then stained with hematoxylin and eosin. This figure is an example of H&E stained tissue from normal mammary tissue in both dogs and humans, as well as an example showing the overgrowth of cells. In the top row, pictures A and B have very neat structure, with epithelial cells nicely surrounding the ducts, and then the stromal supporting cells, those epithelial cells. So in the tumorous tissue seen in C and D, you can see that there's a complete loss of organization and any structure in the mammary tissue. Now it just looks very chaotic. So this is an H&E stained portion of our tumor 111. You can see that there is an overgrowth of cells here, which is an indicator of tumor growth. So immunohistochemistry uses the same tissue of the tumor to detect expression of cancer-related proteins. This is another example from our tumor 111. This is showing an example of estrogen receptor in the tumor. So for immunohistochemistry, the brown stain indicates the presence of the estrogen receptor alpha protein. The tumor is thinly sliced using the same process as H&E staining, but only hematoxylin is used as a counterstain, just so you can see the actual structure of the tissue. And this was done by sending out a portion of our tumor to Baystate Lab, where they used a staining process to see the antibody bound to the antigen, which in this case is the estrogen receptor protein, by linking a secondary antibody and enzyme to it, which has a reaction and stains it brown so it can be seen. So this is a picture showing H&E staining of our cell line 109, as well as an IHC staining to look for estrogen receptor alpha protein expression in 109. There is an overgrowth of cells into the ducts seen in the H&E picture, and there is very little staining for the estrogen receptor seen in the IHC picture. The brown stain appears to just be an artifact since it occurs in an area without cells. So this is how we actually went about assessing gene expression in our cell lines. First, we extracted RNA from the cell cultures, then reverse transcribed the RNA into cDNA using the enzyme reverse transcriptase. This cDNA was then combined with one of the gene-specific primers we designed, along with polymerase and nucleotides, and was then amplified via PCR. We then took the amplified cDNA and ran it on a gel so we could visualize if the gene expression was expressed in those cells. So these are the genes that we chose to look at because each one told us something specific that would help us characterize the cancer. Several of the genes were used to assess whether these cells were more epithelial or more stromal, and the others were chosen because they often play a role in cancerous tumors. Seen in the top 3 rows, which are purple, are our housekeeping genes, which are naturally expressed in all cells, and these genes were used to make sure that our experiment results were reliable. So a main goal of our project was to determine whether or not all 3 cell lines, 109, 111, and 112, varied in their gene expression, which they do. So this is a typical example of a gel run for all 3 cell lines. This gel was testing for the presence of CDH1 and RPS18. CDH1 indicates the presence of epithelial cells and is seen in 111 and 112 and has a weak band for 109. This does actually correspond to the cell culture pictures I showed earlier. 111 was the most epithelial in appearance and shows the strongest band for CDH1. 112 was a mix of epithelial and stromal cells, so also shows a band for CDH1. RPS18 encodes a ribosomal protein that's involved in translation and is used as a housekeeping gene and should be present in all of the cell lines to make sure that our gel results are reliable. And it is seen in lane 11, 12, and 13. The no template control seen in lane 10 is a control that does not have any cDNA in it and should show no band. It is used to detect any possible nucleic acid contamination in the reagents used, but since there is no band in this gel, we can assume there's no contamination. So here are the results for all of the genes that we have tested for so far. These genes were chosen specifically because they would help us characterize whether the tumors were more epithelial or more stromal-like. Estrogen receptor alpha is actually not seen in this chart because it was seen to be negative across all 3 cell lines, but we did not have a positive control to make sure that these were reliable results. So we use several methods of characterizing our cell lines, such as IHC protein detection, cell culture, and gene expression analysis. H&E staining was also used to look at the tumor tissue itself. Each cell line did show differences in tumor growth and cell culture analysis, as well as in their gene expression. We hope to use these cell lines to study human cancer diagnosis and treatment, since dogs are a good model for human cancer, since they share similar environments, development, gene expression, and treatment. So these cell lines may one day be a good resource for human breast cancer studies. So these are all the people who have contributed to the work of the Canine Tumor Project led by Dr. Accaro. I worked with 2 grad students who heavily contributed to the project by performing many of the experiments and developing protocols. Undergraduate Sarah Williams will be doing a follow-up presentation to talk about other research done surrounding these cell lines and tumors. Thank you for listening.